Secondary protein structure. Secondary protein structure is described as localized conformation of the polypeptide backbone, meaning the folding pattern of a protein will exhibit over a few dozen amino acid residues. There are few different motifs that a protein can utilize. First, let's understand that the backbone itself is essentially planar. The peptide band has some pi character due to resonance, since there is a resonance structure where the lone pair on the nitrogen forms a pi band, pushing the pi band in the carbonyl up to oxygen atoms. We know the rotation is restricted around pi bands, so even though it is not a formal pi bands between the carbon and nitrogen, the backbone is fairly rigid, while the sigma band to the R groups can freely rotate. So it is mainly the side chain on each residue that have flexible conformation. We also know that molecules with dipoles or formal charges will attempt to store energy by making electrostatic interactions. And amino acids have such features, so if this backbone can fold in such a way so as to let each residue interact with another residues, it will do so in order to adapt the lowest energy conformation. One way it can do this is by forming something called beta-plated sheet. The backbone will extend one way and then turn back to line up alongside the first part of the chain so that the NH band from one section can form dipole-dipole interaction with the carbonyl of an adjacent section. This stores energy, so it is an example of favorable conformation and one type of secondary protein structure. Another type of structural motif is called the alpha helix. This is when the backbone forms a spiral shape with about 3 or 4 amino acids were torn, and all of the R groups pointing out. Here, it's amine group 3 residues above and the 1,3 residues below. So beta-plated sheet and alpha helices are the most common types of secondary structure, though there are other kinds of coils and loops that are a bit more difficult to describe. The key thing to understand is that at this level, the polypeptide backbone begins to form shapes entirely dependent on how it can best store energy in dipole-dipole interactions by adapting the lowest energy conformation, and this structure contains so many individual atoms that we begin to depict them as colored strips rather than with conventional molecular line notation for practical purposes. Next is for tertiary protein structure. There are a few factors that influence tertiary protein structure. First, residues with hydrophobic side chains, like alkyl groups, tend to be found in the interior of the protein, so that they are not in contact with the aqueous solvent. At the same time, residues with hydrophilic side chains, ones with formal charges or dipoles on them, tend to be found on the surface of a protein, so that they can have dipole-dipole or ion-dipole interactions with water molecules. All of this is just a way for the protein to maximize the electrostatic interaction it can make in solution, which is a big factor in determining the way it will fold. Another motif that stabilizes tertiary structure is something called a disulfide band. We know that a cysteine has SH group called a thiol on its side chain, which is sulfur analog of hydroxyl group. If one cysteine is near another, these triol groups can react with one another by mild oxidation to create a disulfide, which causes a covalent linkage between two sulfur atoms, and therefore can also covalently link two residues anywhere in the protein. This can help maintain the structural integrity of the protein covalent band that are much stronger than the dipole-dipole interaction found in the various secondary structure. Overall, if the protein is highly folded and compact, we will call it globular protein, whereas if it is long and spindly, we will call it fibrous protein. Next is the quaternary protein structure. Some proteins are just one continuous polypeptide chain, but some proteins involve multiple polypeptide subunits that can come together to form a larger protein. These units are not covalently bound. They make only electrostatic interaction with one another, but the interactions are strong enough to allow the subunits to arrange themselves in a specific way, and it is the arrangement of the subunits that determines the quaternary structure of a protein. Like hemoglobin, which consists of 4 totally separate polypeptides arranged in a specific way. A protein is comprised of just one polypeptide, then it will not have quaternary structure. So to summarize, primary structure is just a sequence of amino acids. Secondary structure is the way the chain begins to hold on the localized level. Tertiary structure is the complete holding pattern of the entire polypeptide, and quaternary structure is the way multiple polypeptide subunits come together to form a larger protein. For the second question, which is what is the difference between supersecondary structure and protein motif? For supersecondary structure, it is usually composed of two secondary structures linked together by form or combination of secondary structures that have particular characteristics, shape, and appear in variety of proteins. Examples are beta-herpins, beta-alpha-beta unit, beta-minder, Greek key, and helix-torn helix. While protein motifs are more complex structures created from secondary and supersecondary structural components that are repeated modalities you visualized in many protein structures. Motifs are the certain groups of secondary structural elements and the interactions of secondary structural elements with each other that is giving rise to a fully functional protein. It is a unit of fully functional protein which is having the functions resembles to a proper functions of a fully folded protein and property of folded protein. For the third question is about the different or the characteristics of fibrous proteins, globular proteins, membrane proteins, disabled proteins, and molecular chaperone proteins. Fibrous proteins such as collagens, elastins, and silks are characterized by highly repetitive amino acid sequence that primarily provide mechanical and architectural functions in nature. While globular proteins are spherical proteins and are one of the common protein types, globular proteins are somewhat water-soluble, unlike the fibrous or membrane proteins. Its polypeptide chain intertwine with each other to form a spherical shape. And for membrane proteins, it is proteins that are part of or interact with biological membranes, which includes integral membrane proteins, which are part of permanently anchored to the membrane, and peripheral membrane proteins which are attached temporarily to the membrane via integral proteins or the lipid bilayer. Next is an intrinsically disordered protein or IDP. It is a protein that lacks a fixed or ordered three-dimensional structure. IDPs cover a spectrum of states from fully unstructured to partially structured and include random coils, molten globules, and large multi-domain proteins connected by flexible linkers. And last is the molecular chaperones protein, are proteins that assist the covalent folding or unfolding in the assembly or disassembly of the other macromolecular structure. It is concerned primarily with protein folding. And the last question is an example of denatured protein in our daily life. The common example is when food is cooked, some of its protein become denatured. This is why boiled eggs become hard and meat becomes firm. A classic example of denaturing in proteins comes from an egg whites, which are typically largely egg albumins in water. Another example is a paper clip. When we straighten a paper clip, it loses its functions in clipping papers, like how it was designed and to be used in our everyday life.